Alright, so I'm going to be talking about Sanger sequencing in this video. Basically, Sanger sequencing is a DNA sequencing method that we use for, let's say, we want to sequence something that is short or a little bit medium length size. Normally, I think the maximum size that is really used for is like 1,000 nucleotides only. More than that, usually we would use shotgun strategy or the next level or next generation sequencing. So before I talk about Sanger sequencing, I'd like to talk about this concept because this is the basis of how Sanger sequencing works. If you understand this basis, I think it would be easier for you to understand Sanger sequencing as well. So first of all, if you remember like polymerases like RNA, DNA polymerases, you would remember that it can only elongate if the nucleotide has a free 3' OH. I hope that's kind of like familiar to you. So basically, let's say I have a template, right? This is my template, 5', 3', and then let's say during replication, DNA polymerase will use dNTP or deoxynucleotide triphosphate to add to the growing strand, right? So the 3' end of each one always has a free OH because the OH is really important for adding new nucleotides to the sequence. So if it has a 3' OH, that means the polymerase, whether it's RNA or DNA, they can just keep adding until it finishes, right? So the OH is really important on elongating the sequence. So now, if you remember if I just want to show you like what's the sequence of the normal dNTP. It basically has your 3 phosphates, your sugar molecule, and your base. It could be ATCG. So basically, the 3' OH, this is the important part over here. Basically, like if the molecule has this, that means polymerase can keep adding and adding and adding and just keep elongating until it reaches the end or the next nucleotide that was added doesn't have a free 3' OH. So here's the thing. In Sanger sequencing, we use ddNTP. Basically, ddNTP is dideoxynucleotide triphosphate. It's the same thing here. We just add a di at the first because it's two. Two of them doesn't have an oxygen. So basically in here, this is the molecular structure of a ddNTP. It still has the 3 phosphates. It still has the base. It has the sugar molecule, but it doesn't have the OH anymore. So if there's no OH, that means polymerases like DNA and RNA, they cannot add to it. And then that means the sequence that was growing, let's say it was around here. I'm sorry if it was bad. If the polymerase suddenly added a ddNTP on the 3', that means it has to stop there because it cannot add anymore since there's no free 3' OH. I don't know if this makes sense, but I hope it does. If you're still kind of confused, I would suggest watching the previous video about DNA replication so that you can understand like the difference between using a dNTP and then you can start inferring what's the dNTP. Alright, so let's go back to Sanger sequencing. Sanger sequencing basically kind of relies on this basis or this concept. So basically Sanger sequencing, kind of like PCR, we need a template, a DNA template. And then we also need primers so that, if you remember, primers are basically the strand that tells polymerase where to start, the DNA polymerase specifically. We need the primer. We also need the polymerase so that we can elongate. And then we need the dNTPs, the normal ones. And then we also need the ddNTP. So if you think of it, it's kind of like PCR in a way that it uses everything else except for the ddNTP, right? So that's how I kind of remember Sanger sequencing. It's kind of like PCR, but this time we have to like we need to stop the sequences so that we can start sequencing it from smallest to biggest. Alright, so this is kind of like the required things that Sanger sequencing uses. Alright, first of all, in the lab, when you're doing Sanger sequencing, normally you would have 4 test tubes. Now, why do we have 4 test tubes? Because if the bases that we use are 4, we have A, we have T, we have C, we have G. So basically, in each test tube, we're gonna add everything else. Like, we're gonna add the template, the DNA template, we're gonna add the primer, we're gonna add the polymerases, and we're gonna add the dNTPs. So basically, for the dNTP, we're gonna have a lot of dNTP. It doesn't matter what kind of dNTP it is. Now, to make this more efficient, the type of ddNTP that we would use is normally very specific. For this test tube, we would use ddATP. For this one, ddGTP. This one, ddCTP. And then this one is ddTTP. So the idea here is that we're gonna add everything else, but the ddNTP that we're gonna add is very specific, so that it's easier for us to sequence it once we put it in a gel. I guess I'll show you the gel electrophoresis first, so that it's kind of easier for you to understand. Maybe it's gonna make sense. All right. So once we add everything on those 4 test tubes, and then we incubated, elongation started. Like I said earlier, what happens is that we have this DNA template, right? And then we have the elongating strand. So let's say this is the primer also. If you remember, we can only add at the 3' end, and then specifically we can only add if there's a free 3' OH. So basically, let's say on the first test tube, it was ddATP, right? Polymerase, just keep adding because it keeps using the dNTP that was added on the sample. But it could randomly stop anywhere, and because it could randomly use a ddNTP as well. So for the first test tube, remember we use the A, and then test tube we use T, C, and then G. So let's say for the first test tube we had ddATP. Maybe it keeps going, and suddenly it stopped here. So now we have our first fragment, right? And then let's say for the second test tube, it did the same thing. It kept going, and suddenly it stopped around here. So now, this is our T. We know that there's a T over there. And then to the third test tube, it just kept going, until it stopped here. So now we know there was a C in this fragment. And then for, let's say, the last test tube, it just kept going until the end. And then that's where it stopped. That means there was a G there. Alright, so this is basically like the idea of how the ddNTP works. But we can't visualize this until we do gel electrophoresis. So if you watch the concept of gel electrophoresis, basically we have this gel, and then within the gel we usually put a ladder. Basically the ladder is just for size. It's kind of like like where we're gonna compare the bands that we're gonna see. So for the gel, normally we would set it up where we have a well that is like for A, a well that is for T, a well that is for C, a well that is for G. So because this kind of corresponds with the ddNTP that was used. So if you remember, the DNA is negatively charged, so that means it will go to the positive side, right? So now that let's say we already incubated everything, the test tubes were ready, we're gonna start pipetting it and then put it on each well, right? So let's say on the A well, the bands show up around here, right? Then for the T well, let's say it showed up around here. And then the C well, it showed up around here. And then G showed up on here. All right, I hope you can see that. So the idea here is that the smallest fragment or the smallest DNA fragment will be traveling faster in the gel. So that means this is the smallest fragment because it was the farthest among those. And we would read this from bottom to top, right? So basically, like, if you imagine something like this is our template. The first strand, the very small one, that means we have something like around like this one. This is how short it was. Something like this. And then the second one around here, it was the C. So for this one, we have this was in the A well, right? So we can assume that this one ends with an A. You know what? Let me put a color here. Ends with an A. So that means there's an A sequence on that region. And then the second smallest is a C. So we can imagine that it was something like this again. Something like that. Right? This is the second smallest. And then the third smallest was the T. So that means in this one, we could see something like this. And then the last one, our biggest, is the G. So that means at the end, we can see something like maybe it's gonna be like the longest one. It doesn't matter. I'm just showing you like the idea of how we sequence the DNA. Right? So basically, like, because we know the test tubes has those specific ddNTPs, then we know like when we put it on the gel, we know like where the sequences are, like how big the sequence was. So basically, this one, that means this is the first sequence. The first sequence would have an A. So 5 prime, and then suddenly we have the next one, C, and then T, and then G. Wait. And then G, right? So we can see the sequences, right? But remember, these are complementary to the DNA template. So now if we want to know the sequence of the DNA template that we use, we just do the complementary. So basically the complementary would be C, A, G, T, 3 prime. So this is kind of like how Sanger sequencing works. But the thing here is that these lines over here, we can only infer where the fragment ended. So if we really want to like figure out the whole sequence of the whole DNA template, we will have to have a lot of sizes of fragments, like the very smallest fragments, so that we can figure out the first nucleotide of that fragment, of that sequence. So this is the idea of Sanger sequencing basically. Basically, we just kind of like infer where those sequences are, those nucleotides, so that we can say, oh, it goes from around here. There's gonna be a C here, there's gonna be an A here, the G and a T. Those are the specific places where we can find those. And then if you really want us, if researchers are asked, like we really want to figure out the whole sequence, we're gonna have a lot of or like very multiple sequence, like ddNTPs, fragments, so that we can figure out each nucleotide on each sequence. That's why this is not suitable for very big DNA sequences. So yeah, if I'm gonna summarize this, basically, we can think of it this way. We have Sanger sequencing, right? First, we're gonna incubate it in 4 test tubes. Each test tube has the templates, the polymerase, the primers, and also the dNTP. Now, that's for all test tubes. But to make it more specific, each test tube has a ddNTP that is specific. For, let's say, the first test tube is ddATP, second is ddTTP, ddCGP, and then ddGTP. So basically, you just want to make it specific for each test tube. Incubate it. The fragments will be random. It could be the biggest fragment, or it could be the smallest fragment, the very first nucleotide sequence. And then after that, we're going to view that in a gel electrophoresis. Within the gel, we're just going to observe where the bands are. So basically, let's say for the first test tube, the band was in here. The second test tube, the band was around here. The third test tube was around here. Fourth test tube is around here. So we can infer that the first few sequences of the DNA template would have an A in it. The second will have a C. The third will have a T. The last will have a G. So if you think about it, this is our template. And then if I try to do it here, our first sequence will have an A. The second will have a T. Oh no, a C. And then the third will have a T. And the fifth will have a G. So now if you combine everything, you can basically think that the first sequences are overlapping, so that means it has the same sequences already. So if you rewrite the sequence of the complementary, you're going to have something like A, C, T, G. Right? Now, if you want to figure out what's the specific sequence of the template, we just do the complementary base. Complementary to G is C, complementary to T is A, complementary to C is G, and complementary to A is T. So now we know that the DNA template, the sequence of that, has a C at the very first, A on the second, G on the third, and T at the last. And then, if you really want to figure out the whole sequence, you would need to have multiple fragments of different sizes so that you can figure out like at what specific sequence does this nucleotide appear. So yeah, that's basically how Sanger sequencing works. I hope this video helped.